We are starting with slide 35. We left off talking about isotonic, hypotonic, and hypertonic solutions. So just to make sure, I know I haven't highlighted everything here, but make sure that you know that an isotonic solution means that the cell has been placed into a solution whose solute concentrations are equal. Solute concentration is equal to the solution inside the cell. And in that case, there's not going to be any net gain or loss of water by the cell. The cell will stay the same size. In a hypotonic solution, the solution that the cell is placed into has a lower concentration than on the inside of the cell. I always remember it this way. Hypo, O rhymes with low. Another thing the O does in hypo is it lets me know that the cell swells up like a big round letter O. If you had me in class, I would demonstrate this and look like an idiot. But anyway, that's how I always remember it. It's how I've always taught it and always remembered it. Is hypotonic means the cell is going to swell up like a big round letter O, and the hypotonic solution has a lower solute concentration than inside the cell. In plant cells, this will cause turgor pressure. It will cause the plant cells to swell up as much as they can, but they won't burst because they have a cell wall. But in an animal cell, like a red blood cell, the cell might burst and lyse, which means rupture. Now, protozoans are little single-celled organisms. They are eukaryotic cells, so they're a little bit larger than bacteria. Well, they're a lot larger than bacterial cells, and they have a nucleus and they have organelles. But protozoans that live in fresh water have contractile vacuoles. These are vacuoles that pump water out of the protozoans. And they do this because they continuously take on excess water because the fresh water is hypotonic to their cytoplasm. So they keep pumping water out so they won't swell and burst. All right. Then we have hypertonic, meaning that the solution has a higher solute concentration than inside the cell. So, for example, the cell being placed in a very salty solution is going to cause the cell to shrink. I always remember hyper because hyper kind of rhymes with higher. Now, in an animal cell like a red blood cell, crenation occurs when the cell shrinks because it's placed in a hypertonic solution. In plant cells, the process that happens is called plasmolysis. And you got two examples here when a red blood cell shrinks because it's been placed in a solution that has higher than 0.9% sodium chloride. 0.9% is normal saline. It's the concentration that's in IV. If you get an IV fluid, I'm sorry, I just lost my train of thought. If you have an IV and you're getting fluid because maybe you're dehydrated, you're in the hospital and you're dehydrated, or they're using an IV to get some kind of medication into you, such as an antibiotic, that IV solution is going to be 0.9% sodium chloride or 0.9% saline. So if your red blood cells are surrounded by that 0.9%, they're not going to change size. But if the solution is higher than that, your red blood cells will shrink and they will die. They will undergo crenation and they will die. And plant cells, when they undergo plasmolysis, they also die. Okay, so facilitated transport is diffusion that occurs through a carrier or channel protein. Glucose, which is a simple sugar. It is the sugar we call blood sugar because it's found in our blood. And amino acids and ions like sodium and potassium ions that have a charge. So an example of an ion would be a sodium, forget that. But it's just Na plus, or potassium is K with a plus charge. That's a K with a plus charge, or even negatively charged ions like chloride ions. Any of those that have a charge, or even a molecule that has a charge, any of those are going to have to use a carrier protein to move, or a channel protein to move across the plasma membrane. And they're still moving from high to low concentration, so that means with or down their concentration gradient. So there's no energy that is required. This is a passive process, but it's called facilitated transport. And here we have a solute molecule. Here it is in green. Its high concentration is outside the cell and its low is inside. So it's going to move from high to low through a carrier protein with no ATP. With no ATP required. That is facilitated transport. And this shows you the process kind of as it occurs. So it's high concentration moving through the carrier protein into the cell, into its low concentration, and no energy is required. Active transport, and just a hint, this is one of your lecture quiz, your Chapter 5 lecture quiz that's after these recorded lectures. Ask you about active transport, ask you specifically about the sodium-potassium pump. So active transport is like facilitated transport, but just picture that the molecules are moving from their low concentration to their high concentration. And if that happened, then ATP would be required, and that would be an active transport process. The sodium-potassium pump is probably the most common example of active transport that we use in biology, because almost every cell has sodium-potassium pumps in the plasma membrane. So the sodium-potassium pump uses ATP to move 3 sodium ions out of the cell for every 2 potassium ions that it moves into the cell. This would be against their concentration gradient, and it would require ATP. This answer can be used as long as you'll reword it. I don't want you to copy it, but if you will reword this answer right here and put it into the quiz question about the sodium-potassium pump, that is what I'm looking for, okay? That will give you the 4 points that you need. All right, the last type of transport, and we're going to end with this, is bulk transport. Now, if bulk means large, a large amount of material. So if the material is moving out of the cell, we call it exocytosis. And if it's moving into the cell, we call it endocytosis, and there are 3 different types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. So one example of phagocytosis is when human white blood cells engulf pathogens. And an example of receptor-mediated endocytosis is when cholesterol is transported into cells. Okay, so this is a slide showing the sodium-potassium pump. Let's look at this. The sodium are yellow, and then the potassium ions are pink. And notice that the concentration of sodium is high outside the cell, but even still, 3 sodium ions are being moved out of the cell. And the concentration of potassium is high inside the cell, but even still, 2 potassium ions are being moved into the cell. ATP is used to accomplish this. This is your ATP molecule. So we're seeing 3 sodium ions being moved out against their concentration gradient toward their higher concentration, and 2 potassium are being moved in, again, against their concentration gradient toward their higher concentration. This is a picture of exocytosis. You have a solution that needs to be moved out of the cell, so it's closed in a vesicle here. That vesicle fuses with the plasma membrane. The reason it can do that as you can see, is that the vesicle membrane is also a phospholipid bilayer. So they can just fuse with each other, and then the vesicle becomes part of the plasma membrane. It will actually make the plasma membrane larger, and all of that material is now moved out of the cell. That's exocytosis. Picture that happening in reverse, and then you have endocytosis. You have kind of an indentation of the plasma membrane, and then it eventually will surround this large particle here and bring it into the cell enclosed in a vesicle, and that's phagocytosis. Because this is a solid particle, phagocytosis is also referred to as cell eating. And pinocytosis, this is showing you, is a solution. The solute is green, but it's surrounded by water. So it's being brought into the cell by the formation of a vesicle, and that is pinocytosis, or cell drinking. And receptor mediated is really the same as pinocytosis, but it's just that the solute particles are binding to receptors when they're brought into the cell. So the vesicle is coated, and it contains these vesicles that bind the particles that are being moved into the cell. So receptor mediated endocytosis is very specific as far as what it brings in. And I'm going to let you go through section 5.4 on your own. There is some information that you might need to know, but the most important sections are the first three.